All right, first and foremost, I would very much like to thank the organizers of this meeting for inviting me to give an address at this time. We're going to talk about hormones of the holobiont. These are signal compounds that act between microbes and associated plants, and discuss these in the context of their capacity to be new technologies for more sustainable and climate change resilient agriculture. There is a pressing need for enhanced food production. Growing global population. We're headed, most projections say for somewhere between 9 and 10 billion towards the middle of this, well, towards now, maybe a little after the middle of this century, I think is what they're saying now. And on top of that, there have been changes in diet, so we're consuming more meat, and the additional meat consumption means we need to produce more crops to feed those animals. In addition, there's the threat of climate change. We have the usual things going on. Sadly, they've become usual. We're becoming accustomed to them. Melting of mountain glaciers, the Greenland ice sheet, the last few years has been melting across much of its surface. We're losing Arctic sea ice, consolidated Arctic sea ice at an impressive rate. For here in Canada, we're talking increases projected at something like 5 to 8 degrees by the end of this century or so, if we don't do anything about it, with probably 5 degrees at lower latitudes and 8 degrees at higher latitudes. Actually, let me put my laser pointer on here. That's nicer. In addition, we're seeing birds return sooner in the spring and flowering sooner for plants. And we're also seeing more extreme weather conditions globally, but certainly here in Canada as well. We're seeing more drought conditions more often, high temperatures, salinity where things like drought become more common, flooding from time to time. So all of these things are becoming issues. So one of the things we're considering is biomass for advanced biofuels and the associated bioeconomy. A key step in this is the supply of a reliable supply chain, which brings high quality biomass. This can be crop or forestry residues, purpose grown materials such as fast growing grasses, plantation forestry, things like that. Sorry. In addition, we look at biomass production, so advanced biofuel biomass production on marginal lands. This is to compete minimally with food production. So we don't want to be, given what's happening with food availability, we definitely don't want to be competing with food production. It tends to more frequently stress, though, if it's on marginal land, because the soils are often shallow, there can be water availability issues, nutrient limitations. So we need technologies that allow more production on these soils, make them more productive overall. And if it makes food crops more productive, there'll also be more crop residue. So what you're looking here is food and fuel as opposed to food versus fuel. More advanced biofuel production overall, so more reduction in greenhouse gas emissions, CO2 in particular, from fossil fuels. So this is what we're hoping for. The phytomicrobiome, plants, like mammals, have a microbiome. So it's the phytomicrobiome because this is with plants. It's present through all terrestrial plant evolution, or has been present through all terrestrial plant evolution. The community of microbes are associated with pretty much all plant parts. So you have a group with the roots. In the roots, they're in humid soil. There's organic matter in the soil. So this one tends to be the most complex and the most well-developed in many ways. But they're also in the stems, in the leaves, and even in developing reproductive structures. So they can help in a wide range of ways: nutrient mobilization, production of hormones or hormone analogs, disease control, so biocontrol, signals, so hormones of the holobiont. I'll come back to this. We mentioned this at the beginning. Adapting to climate change, developing inputs that adapt agriculture to worsening climatic change conditions, improve plant stress tolerance, climate change resilient crop production, and in the end, reduce N2O production. So let's say you're dealing with something that helps increase nitrogen fixation, so that would help, and reduce CO2 emissions. So these are the things we'd like to see happen. And if you're doing materials that can be used as biofuels, can help with that, or material that can be sequestered into soils to increase carbon. The approach overall, say biological inputs, addition of small amounts of inexpensive material can enhance crop growth. And this is will reduce costs if it displaces some chemical input, and enhance stress resistance. So this is what we're hoping for: abiotic and biotic. I'm going to talk mostly about abiotic here, but just to show a little bit biotic. This is the same strain in two different places on a Petri plate that's been streaked with a bacterial pathogen. a plant pathogen, and you can see that this is working against this pathogen. This is the control. It's working pretty effectively. This was part of an exercise we carried on. This is data from that. We last year we published the structure of a compound, an isoquinoline ring with various and sundry side chains that works against tomato canker pathogen, Clavibacter michiganensis. So there's lots of interesting things that can be done in this area. I mean, in the bigger picture, nitrogen-fixing inoculants for legumes have been around for over a century now. There's various microorganisms, bacteria that are involved in this. So far, only prokaryotes are known to fix nitrogen. They've been widely used, as I said, for over a century. There are also microbial mycorrhizae inoculants that have been assisting plants with phosphorus uptake, and they've been available for, I guess, a few decades now. And there are bacteria that will help with mobilization of things like phosphorus and zinc. So these things all help. So this is just, I'm going to go through an example of a line of research that actually constitutes quite a chunk of my career. So it started with soybean. When I came to McGill, soybean had just started to be cultured in Quebec. So the thing about soybean, it evolved in the tropics and it was domesticated there. Probably sort of south coastal China seems to be what most people think, but in a warm environment, and it carries that evolutionary baggage with it. So we agriculturalists have carried it further north and further south ever since it has been domesticated. Its metabolism is adapted to and set up for relatively high temperatures. It's been known for over a century now. I think that the optimum temperature for soybean nodulation is 25 to 30, and I'll show you in a few minutes that that's not what we get in soils here in Quebec in the spring. Production in Quebec has, as I said, been since about the time I came here, which was actually the mid to late 1980s. So, but this is something new. So we started growing soybean here, and we observed that when the soybean plants came up under field conditions, they would look nice and green when they come into the ground, but then as they exhausted the protein in the seed, which contains nitrogen, they began to look nitrogen deficient, pale green. And after a time, after they were pale green for a while, they regreen. But this suggested that their growth was nitrogen limited for a while. And we wondered if this could be due to inhibition of the nodulation process by low soil temperature. As I said earlier, that the best temperature for nodulation was 25 to 30. And if we look here at seeding time, we're often down around 10, 12 degrees. So yeah, real potential to be inhibitory. So we were able to show that the temperature slowed the onset of nitrogen fixation. And at the beginning here, as it's dropping, for every degree drop, it slows it by about 1 to 2 days. Once you get below 17, it's sort of 7 to 10 days. And once you get down to about 10, it just doesn't happen. So the cause of this disruption in signaling during the first stage of nodulation was not clear, but we were lucky we were able to do some testing. We showed that indeed it was the very beginning of nodulation was affected. And we were reading the literature. They were just publishing at that point what was going on in the first 12 hours or so. And in that sense, we were quite lucky. So what's going on is the plants are emitting signal compounds. These are isoflavonoids, which trigger genes to come on inside the bacterium. These produce a signal compound back, a lipoketooligosaccharide, back to the plant, and these turn on a set of genes related to nodulation in the plant. So this was lucky for us. So we developed an effective solution to this. We just added the appropriate isoflavonoid signal for soybean genistein. There's a couple of them work, but we used genistein because it was least expensive. We added it to Bradyrhizobia japonicum cells before applying them as an inoculant. Turns on the nod genes, we let it sit for a while. Turns on the nod genes, they start making, they return LCO signals, and all is good. The B. japonicum cells then begin to produce and excrete these LCOs. And by the time you put it on as an inoculant, that part of the problem or that part of the process is out of the way. So this had an effect on nitrogen fixation and growth. So here we can see June 17th sampling. This was a number of years ago. We're just starting this work. But the nitrogen fixation started 4 to 5 days sooner, and the total amount of nitrogen fixed was a prodigious increase, 80%. So not trivial. And yields were increased. So we saw yield increases as high as 40%, but often they were in the 10 to 20% range. But pretty nice, all in all, quite good. This resulted in intellectual property. What happened was I filed a report of invention on this with McGill University. This results in a patent search, patent filing, usually the company associated, if there is a company associated, they pay for this. And it's because it's expensive. They're looking for novelty and utility when they're looking for patent, when they're considering patent filing. The cost is about $15,000 Canadian dollars for Canada and the US, and about $150,000 for a global patent. So it's not cheap. What we did in our case, we set up a spin-off company. It's called Bios Agriculture Inc. And I guess there's a variety of reasons why you can do this, but the reason I did it at the time was because I had a technician who was a very good technician, and I was grant money was getting hard to get at the time because we were just at the end of a recession and the effects of various budget cuts were trickling down all the way to the lab. And so I wanted to keep this technician employed post-recession. So he and I started a spin-off company out of McGill, and the whole thing with spin-offs was a bit new at McGill at the time. So McGill took a 5% non-dilutable share, which was interesting. Again, we didn't know anything about these things or what they meant at the time. And then there were the usual other things. We were dealing with venture capital and other investments. And then there's regulatory requirements of government that you have to go through. They have to be satisfied before you can market a new product. But anyway, we soldiered on and got through most of this. As is so often the case in research, when you start doing something, you trip over things. So we were testing in the field, and one of the interesting things we observed at some of the sites was that they would come out of the ground faster when the genistein was applied to the bacteria, so they were making the LCO. So what did this mean? One of my grad students and I designed an experiment. We said, okay, look, we'll test, we'll just do a germination experiment. We tested seeds in water, genistein added to the water, genistein plus the seeds, and then the Bradyrhizobium japonicum added by itself, and then the genistein plus the Bradyrhizobium japonicum, which would result in the production of LCOs. And when we did that, it was the last one, the material that made the LCOs, that actually caused the increase in seed germination rate. So it was pretty interesting. And what was also interesting on top of that, when the student and I designed this, at the end of it, we were just finishing up, and I said, you know what, try this on corn too. Corn has a serious problem with soil temperature and germination. And you know, it doesn't seem likely it would help, but maybe it would. So we tested that, and sure enough, it worked on corn too, and we'll come back to that. So here we are. So it worked on in fact, it worked on a whole lot of other, like this is the control. Here we can see the LCO treatment for corn. It's pretty dramatic difference. Also worked on cotton, beets, there's soybean again. It worked on pretty much everything we tried it on. So we commercialized this. Bios Agriculture, as it was called, was eventually taken up by Agrobiotics, which is a larger Canadian company. There's a kind of a corporate food chain happened here. So Agrobiotics, yeah, it's purchased by EMD, which was the largest inoculant company in the US at the time. EMD actually, I would say, became, it's maybe not the very best way to say this, they sold part of themselves, which was then folded into Novozymes, and that contained our technology. So EMD then had our technology, and then on to Novozymes. So Novozymes actually eventually hardwired itself to Monsanto, and Monsanto was swallowed by Bayer, and so it goes. Okay, this is just some data you can see. This is the first time I'd ever seen a large-scale field study, that the kind of thing industry would do. This is 37 sites, site years, as it were, across in 1 year, but across 37 sites at disparate locations. And you can see there's a mix of, I guess, basically noise down here at the bottom, and then some big pluses. The person who showed this to me was from EMD. They were very happy with this. Here's some other data, all showing positive effects. So they were pleased, and needless to say, we were pleased. But there was also some other considerations in all this, or there were some other considerations, and we were looking at other things. Flavonoids have two problems. They can be very expensive. As I said, we used genistein because it was the cheapest, but it was still expensive. And they're often used as phytoalexins and things like that by plants. So they can work against biological entities. So at 20 micromolar, which was the concentration we were using, they could slow the growth of the rhizobia, which was already slow. So that could make it more expensive producing the inoculant. One of my grad students found a paper that suggested jasmonic acid might also activate nod genes. It's a lot less expensive. In fact, I think when we were doing this, a bottle of methyl jasmonate, a quarter-liter bottle, as I recall, was $60. So very inexpensive. It's not damaging to rhizobial cells. Interestingly, it's generally involved in stress-related signaling, and we'll come back to this. So it's both involved in potentially biotic and abiotic stress management. And in this case, the signal to the microbe that the plant is stressed, and the microbe responds with a signal to manage stress in the plant, presumably. Anyway, I'll explain this more as we go ahead. So both jasmonates and flavonoids are stress-related. So the findings in general, jasmonates induced expression of NOD genes in Rhizobium japonicum. They also induced the return LCO signal production. However, the strain specificity was very different from the isoflavonoids, and they accelerated nodulation at low temperature in the root zone, which is great. That's what we wanted. And we commercialized this. So here we are, jasmonate effects. It took higher concentrations to get to it, but it's so inexpensive, it didn't really matter. And it actually achieved higher level of expression than the Genesty. So all good. Here we are looking at some effects in the field. This is the control. This is just inoculation, so you see an effect there. And here's the effect of genistein being added. Here's the effect of jasmonate being, or methyl jasmonate in this case, being added. Here are the two together, so no real indication of synergy there. Okay, this was an interesting development. Yeah, so we licensed this technology to Becker Underwood, a company called Becker Underwood. And Becker Underwood started marketing it pretty intensely, taking a bite out of the Novozymes market share, and Novozymes didn't like it. So Novozymes sued Becker Underwood for patent infringement, and Becker Underwood countersued, and all the patents were out of McGill, out of my lab. So I got to spend most of a year in court doing depositions, etc. But in December, a Wisconsin judge dismissed both suits, which I was very grateful for. BASF bought Becker Underwood, and McGill audited Novozymes, and actually it turned out Novozymes was not paying what they should have been on the royalties. So McGill did well by that. There's some work on other PGPR, plant growth hormone rhizobacteria. I had a grad student at the time that isolated 14 strains from surface sterilized nodules of particularly vigorous field-grown soybean plants. So these are plants in the field that they were doing really well, and there was no obvious reasons why they would stand out from the rest. So maybe it was because of associated microbes. So we isolated microbes from their roots. The microbes were tested for ability to promote plant growth under greenhouse conditions. 3 were particularly effective. So, and this one, NEB17, just 17 because it was the 17th tube in the rack. Nodule endophytic bacteria is what that stood for, at least initially. And growth stimulation effects. So plant growth effects were tested on plants grown in growth pouches, in pots, and in the field. And they all had effects on these in some cases, but again, the NEB17 was the only one that worked in all 3 cases. And it caused some very nice increases in yield in the field. So this is the kind of thing you see: increases in nodule weight, increases in nitrogen in the plant, and increases in the yield. So it's very, very, very gratifying, very hopeful in terms of something that could be commercialized. We had done some work also in some other PGPR signals. We continue to do it, and this is just a little bit about our general approach. We'll produce the cells in a liquid culture, then we'll remove the cells often by centrifugation followed by filtration, and then we'll test the resulting cell-free supernatant and make sure it still has biological activity. Then we'll run it through an HPLC, and what we'll often do is we'll divide our HPLC run up into, in this case, quarters, and we'll see which one has activity in it, and then we'll take that quarter and divide it up into quarters, and we just keep going until we have a single peak. And then you can start to get, there's testing to be done. I mean, the peak may still have more than one compound in it, et cetera. There's things to do, a lot of chemistry. So then we looked at the signaling more closely, the NEB17 signals. So NEB17 produced thyrosin 17, or at least that's what we called it. It's actually now being called bacillin 20. I could explain that if somebody wants to know. Stimulated seedling emergence and subsequent plant growth. So it also turned out to have medical applications because it's a bacteriocin, and similar effects. Well, this is another protein we're working with. We haven't followed up with this one because it's hard. It's produced in smaller quantities, and it's just harder to work with. So. Corn, okay, I said before it had effects, potentially some of these things have effects potentially on other plants. That proved to be the case with thyrosin 17. It showed effects on corn, which is a C4 monocot, and soybean, a C3 dicot. If it's showing positive effects on two such diverse plant types, then it suggests that most plants will be affected, and that's been our experience. We were trying to get a sense of what it was. Actually, initially, I thought it was probably something like the LCO, some sort of chitin-based material. But when the student who was involved was trying to find that out, she tested it and said, you know, I'm not getting any activity for chitin here, so I'm not sure what's going on. So we did testing, and eventually, with staining, it lit up for protein, and then here we are. This is stuff coming out of our HPLC, and you can see this is here we are showing indications of protein. So quite a lot of it. The fact that it's a bacteriocin, it's one of those things that we were trying to figure. I had a master's student say she was very good, and her whole thesis was just really, what is this? And she called me up late one night, I think about 11 in the evening. She was all excited. She's going, I think I know what it is. I'm going, okay, what do you think it is? She said, it's a bacteriocin. I go, well, that's nice. And I said, what's a bacteriocin? I didn't know. So anyway, she explained it to me, and this is the thing you see here. This is we tested it against 53 strains, and it inhibited 32. So that's usually it inhibits more closely related strains. And you can see here, you can see the inhibition zone here. This is on a different media, so it's a different color. It's an inhibition zone. Another media, another color, inhibition zone. And here's one with no inhibition zone. So no inhibition here. We worked on the gene sequence. We did a collaboration with another lab because we were doing very much genetics work at the time. So in NEB17, interestingly, we found that it had 3 variants of the gene, but they all coded for exactly the same protein. They had been separate long enough that they had evolved a little bit, but where there were differences, they were usually in the 3rd position, the 3rd in the codon, and they always coded for redundancies. So I think what's going on here is this protein is doing 2 things for the microorganism, for the bacterium. It's both expanding the niche space by making the plant grow better, at least under stress conditions, and it's also clearing the niche space of the closest competitors. Bacterioses often work on closely related bacteria, so it's clearing the niche space of those guys. So more niche space and less competition. So it's doing 2 things. So in an evolutionary context, if it's going to be changed, so it increases one of those activities, it has to at least not decrease the other. So it's an interesting situation. And I say we did a predicted sequence that matched at least our partial initial amino acids. We started sequencing and we could get a ways in from each end, and then it would stop. It may be related to disulfide bridges or some other things. We're not sure yet. Predicted molecular weight matched the measured weight, and the gene included an N-terminal extension that could be related to moving the mature peptide out of the cell. So here we are. See, 3 repeats, the 3 types, somewhat different in terms of nucleotide sequence, but all have coding for the same protein. So again, the dual function protein, or actually maybe more, given the size, it may be more appropriately called a nucleotide. So environment and temperature response, this was the thing. So in the lab, and I kind of alluded to this already, responses could be variable, and that could be frustrating. In the field, the responses were, we noticed the strongest for us at this latitude, cold springs. We have them often, heavy soils where the soils retain water and stay cold longer. This is where we get the strongest responses. So we started to wonder if this was related to stress. And this is a Arabidopsis. So 22 is a really good temperature for Arabidopsis. You can see the control is in the middle here amongst these various treatments, not much effect. And this is Arabidopsis at 10, which is stressful for it. And you can see here, the control doing not nearly as well as the treatments with signal compounds added. This is with Versin 17. Actually, we also got LCO in here. We couldn't resist testing them both at the same time. So, and this is just more indication of this. This is drought stress. Here we are. This is no sodium chloride. This is, sorry, drought, so salt and drought. This is salt. Sodium chloride. Here we are, 150 millimolar over here, and here's none. So this is the control for this. This is the control for salt by itself, and you can see the salt has made the plant about 10% the size of the 0 salt plant. But what we also see is adding the LCO at the correct concentration, and it's interesting you should note the concentrations here: 10 to the minus 6 and 10 to the minus 8 molar, and the Versin 17, 10 to the minus 9 and 10 to the minus 11 molar. It works all the way down to here, and it produces quite a lot of it. So this stuff is easy to make lots of. And this is a polyethylene glycol. So this is sort of water stress. It's an osmotic stress. So this gives you a sense of how it could work against drought. And here we are. Some other indications of this. We see increases in the levels of stress response proteins when it's added. And anyway, this was sort of backing up the notion that it was helping the plants deal with stress. Soybean nodulation, we saw increases indoors in plant biomass, root biomass, even bigger. Nodule biomass, nitrogen in the plants, and under field conditions, the only thing related to nitrogen fixation we measured was nodule mass at flowering. We saw nice increases there. Yeah, okay. So, when it turned out to be different chemically from the LCO, that is Tyrosine 17, I guess that they were binding to the same receptor. I'm not at all sure about that now, because if you look at the changes in the hormone profile for the plants, here we see that, some of them, it looks similar at least, but some of them it's very different. And so you'll just go, I don't know if it's working through the same receptor. It's having some pretty remarkably different effects, at least initially. In the end, there were increases in stress tolerance and increases in growth, but I don't know. And this is a more gene responses. These are soybean plants. They're being grown at 15 degrees. So that's stressful for soybean. And we measured changes in gene expression when we added the LCO spray, in this case, or back to the LCOs. And interestingly, what happened was the class of genes where there was the biggest change in expression were stress response proteins, which to me, this is very interesting because these plants were already stressed. So it seemed to suggest they're going from one set of stress response proteins to another, which, it's an interesting situation when plants are stressed. They kind of have two choices. They can try to grow through it, which as agriculturalists or bacteria that are trying to live off the plants is what you want. Or they could become sort of quiescent and begin to shed tissues and things like this and just try to keep the meristem alive and get through it. And it's not really what you want as an agriculturalist. So this may have been what's going on. It may have been shifting into the grow through it strategy. I don't know, but it's an interesting situation. This is looking at sort of overall expression of genes and proteins. And you can see that this is actually proteomics, but you can see that a number, we already saw changes we knew related to stress tolerance, but here we see a number in key energy related pathways. So some of the other things that went with this, we tried it on some this approach on some biofuel feedstock materials. We isolated growth promoting rhizobacteria and tested them. The 3-year experiment, both LCO and Tyrosine 17, we found amongst the treatments increased plant growth and development. In a 1-year experiment, we selected plant growth promoting rhizobacteria, as I said, and this increased yield by up to 40% in the establishment year. So these are all good things. More recently, we've been doing some work on cannabis, and we had some novel strains from phytomicrobiome plants that were isolated literally out of my backyard. We screened them for plant growth promotion, specific beneficial traits such as nutrient mobilization, etc. We inoculated the best 3 on a range of plants, including cannabis. And for cannabis, we found that it increased flower yield something around up to 11.5%. They increased THC and CBD both at the same degree, or I think proportional increase, up to 14%. So you get a combination of increasing flower and then increasing concentration of the cannabinoids in the flowers. So it seems very interesting, and there's some suggestion, at least, we have indirect evidence that suggests there's interorganismal signal involvement in all this. We're going to look for those next. It's also a strain from a non-cannabis plant that's being patented for strong activity against one of the more important cannabis pathogens. So that's a biocontrol approach. So what they are, again, these signals, we have the holobiont. This is the combination of the phytomicrobiome plus the plant. And so I've started referring to these in some of our papers as hormones of the holobiont. They work at very low concentrations, as we showed above. They're clearly not metabolites, so effective at very low concentrations. They regulate plant activity in a very meaningful way. You get large changes in gene expression, protein production, hormone profile, many other things. They can regulate other microbes, which could then regulate the plant. We haven't done work on that. They constitute a powerful way to affect plant growth and productivity. They're inexpensive to produce and apply, low environmental impact, in part because there's a small amount, sorry, I just went on, already present, and they're already present in the, sorry, they only require a small amount, and is already produced in the environment, so they are unlikely to be a problem environmentally. They're poorly understood, no question. These signals are, I'm sure there's lots and lots more of these to work with, and we just don't know about them very well. This enormous potential, I've been asking myself recently if we should try to form some kind of national or international network around this. And I guess right now we have, well, actually we have quite a few stories. We had more than 10, I think actually at this exact point, we have quite a few more than 10. Yeah, so the phytomicrobiome potential, wide range of species from the phytomicrobiome, they're there now, and we can draw on them and screen them, etc. The most powerful control factor is the plant. It seems to be able to, sorry about that. It seems to be able to exert effects on what types are there. So the development, stress level, nutrition, all of these things, whatever the plant is experiencing, it regulates the strains present to go with that. They seem to be key to plant growth and productivity, and especially under challenging conditions. Management, you can inoculate them. You could just try to inoculate key ones and manage what are called hub species that manage other microbes in and around the roots. And, just say signals. There's microbe-to-plant, plant-to-microbe, microbe-to-microbe. All of these could be important. And this, again, this is a Arabidopsis plants, and you can see here, this is again salt. Here's LCO, 2 different concentrations, tyrosine 17, 2 different concentrations. So again, showing some remarkable improvements in growth. So if you're going to do these things, you're going to isolate them, work with them, develop them. Who would be involved in this? It takes a bunch of players, and then you need to have collaboration amongst them. So basic research labs could be academic or government, and industrial labs, and then after that, you need maybe other aspects of government and industry. But anyway, here we are. This is some of the ones that we've been involved with. The overall approach, sample plant-associated microbes. So this is the phytomicrobiome. Conduct efficient, rapid screening. We've gotten quite good at this, so we can do it pretty quickly now. You do the biological assessments, then you do the agronomic assessments. And so finding and screening the microbes, where to look. Collect microbes that can be cultured. We're stuck with that. That's all we can do right now. And it's only about something maybe around 5% of the total. So you can collect these from crop plants. The microbes associated with crop plants are not very well understood. As in the roots, in our case, that's mostly what we're focused on. And the signals that are going back and forth between those microbes and the plants are extremely poorly understood. You can look at specific resilient wild species, and you can look across a range of habitats. The interactions between plants and their associated microbes for undomesticated species are even less well understood than for crop plants. Then you do efficient, rapid screening for early growth response. So we do seed germination initially. Let's get some that look promising. We'll try them seedling emergence, then early seedling growth. We can measure photosynthetic activity. We can carry the plants along a little bit and look at gene and protein expression. You wind up with single strains. Some of them might have a range of benefits, so you could combine them into consortia. All kinds of things that can be done here. Agronomic assessments, field experiments, range of soils and climates. And it really is important. Generally, you want to do a set of tests across most of the area where the crops you're interested in might be produced. Planting dates, reasonable geographic area, reduced inputs. One of the things we'd like to be able to do is use these to reduce the chemical inputs at least somewhat. You have to get through the regulatory process, get on to full commercialization, but the potential is very large indeed. This is just some of the folks. I've really just summarized about close to 40 years worth of research. So it's this is just happens to be a random sampling from a few years ago. I had this picture. So that's some of the people, but there are many of them. And so that's the end. And thank you very much. And again, I want to thank the conference organizers for inviting me to do this. Take care. And if you have any questions, you can email me. Actually, my email address is donald.smith@mcgill.ca.